The Coronavirus in the Urban Built Environment Project, or CUBE, began with one simple question. Can we find SARS-CoV-2, the virus that causes COVID-19, on surfaces in the built environment? As it turns out, yes, we can. When we say built environment, we're talking about basically any human-made structure. For example, a hospital, a school, or a long-term care home. And we're also talking about the features and objects within that structure. Tables, chairs, countertops, elevator buttons. Surfaces in these built environments can host viruses and bacteria. Don't worry, it is extremely uncommon to get infected with COVID-19 from any surface. But what these surfaces can do is tell us whether the virus is present in that environment. And through our prior research, we've discovered that the most reliable surface on which to find the virus is the floor. When a person who is infected with COVID-19 breathes, speaks, coughs, sneezes, the virus travels outward in those respiratory particles. It remains suspended in the air for a time, but eventually falls and settles on the floor. This is where CUBE comes in. We use cotton-tip swabs, just like the ones used to test people. Except instead of someone's nose, the swab is rubbed repeatedly across a small section of the floor for 30 seconds. It is then stored in a stabilizing solution and transported to a lab, where it undergoes a type of test called PCR. PCR tells us if the swab sample is positive for the virus, meaning the virus has been detected, or negative, meaning the virus has not been detected. PCR can also help us estimate how much virus is present in each positive sample. So, we can tell if the virus is on the floor, and we can estimate how much virus is on the floor. But what can we do with this information? Through our research, we have learned that the presence and amount of virus on the floor correlates to the prevalence of the virus in human beings in that environment. The more positive swabs we find, and the greater the amount of virus in those positive samples, the more likely it is that COVID-19 is actively circulating in human beings in that environment. We can use this as a signal to predict when and where an outbreak is about to occur. And through continued sampling over time, we can assess whether the outbreak is improving or worsening. How? Well, as the size of an outbreak increases or decreases, so does the amount of virus we find on the floor. The more people infected in an outbreak, the more virus we find. The fewer infected people present in the environment, the less virus we find. Another key piece of information we get from sampling floors is localized detection to specific areas within a building. For example, positive samples might be predominantly found in staff-only areas, or perhaps only on specific floors of the building. Based on swab positivity and the location of positive swabs, organizations and individuals can then make decisions about infection prevention measures to stop the virus from spreading. Measures like masking, distancing, human-based nasal swab tests, visitor policies or restricted access to certain areas, and other measures that help keep people safe until an outbreak or the threat of an outbreak has passed. The CUBE project aims to further develop this approach of sampling the built environment as an early detection and monitoring system to help protect all of us in the spaces that we live, work, and play. To learn more about the project or to contact the CUBE team, please visit our website at cube-ontario.github.io.